genetic and genomic variation. So this chapter is going to focus on principles from more of a population genetics and to explain some genetic and genomic variation as it applies to populations and not just individual patients. This will help the student to understand the implications of principles such as founder effect, genetic drift, and population bottlenecks, as well as applying those variations in populations with which they are familiar. We're also going to talk a little bit about the Hardy-Weinberg equilibrium and the ways in which populations change or stay over time and due to certain circumstances. We'll also talk about common references and understandings of the terms of race and ethnicity and the lack of a clear biological basis for such groups, even though they remain culturally important and contribute to our understanding of health disparities. So, first of all, humans are very homogeneous. We're 99.9% alike. How we vary is individual DNA sequences, copy numbers, polymorphisms, and chemical additions that result in differing levels of a gene expression, epigenetics. So mitochondrial DNA and DNA on the Y chromosome is passed from generation to generation with very little variations. Factors that increase diversity. Genetic diversity is due to independent assortments of alleles during meiosis and crossing over of segments of homologous chromosomes. So we have this independent assortment of alleles, and during meiosis is when that diversity takes place. Genetic anthropology uses genetic information and physical evidence to learn more about our history. National Geographic Society's Geographic Project looked at DNA samples from all major populations and cataloged all those genetic similarities as well as the differences, and they have an open source database. So it's this huge database on DNA samples from populations from all over the world, and it's cataloged so that when we cross-reference different DNA and see the human genetic variations. It's pretty interesting. There's a video on Blackboard that addresses some of this, so be sure you watch those videos that we posted. Haplotypes, excuse me. These are genetic neighborhoods, so to speak. It makes it easier to study the differences as far as the risk of disease and response to drugs that found in different populations. So groups or genes or SNPs or markers that tend to be inherited together are haplotypes. The International HapMap project was completed in 2005. It's an international collaboration looking at the genetic neighborhoods, so to speak. Population genetics examines the way in which the allele frequencies change in human populations over time. So what events keep these frequencies the same and what events change those frequencies over time? This is what we look at if we're doing more population genetics. A species with a lot of different alleles will have more genetic diversity, which totally makes sense. Geographic origin of ancestors. So this is what people are kind of interested in right now. If you see there are different advertisements where you can send in your DNA and have that, and they can match your ancestry, which I think is pretty interesting. The rates of disease risk that vary by the geographical origin of ancestors. So like with sickle cell disease and hemophilia A, how those vary according to population, migration, and different geographical origin. There's also natural selection and migration patterns will also affect the way disease rates of disease risk. So the notion that organisms are best suited to the environment, those will survive and reproduce and pass on their genetic characteristics. That's what natural selection is. So that plays into how different rates of disease occur in certain geographical areas and within certain populations. Allele frequencies, one sequence variation for every 200 to 500 nucleotides, changes that allow us to adapt to extreme environments, changes in our gene pool. And gene pool refers to the sum of all the alleles for the genes in a given population. Evolution is a change in the genetically inherited frequencies of alleles over time. So that's the technical definition of evolution. Heterozygosity refers to a proportion of the population who are heterozygous and at a particular locus. So how many people in that group are have the certain alleles and the certain makeup or genetic makeup. So if 95% of the alleles in a population are dominant, then most of the people will be dominant trait. The heterozygosity will be low. But if you have a few that are either non-dominant or you have one dominant and one non-dominant, then you know that heterozygosity would be higher. So it's just a way of looking at the proportion of your population in terms of how the alleles are divided, or who's got how many people in one section have how many traits. That was a really long explanation for a pretty simple concept. Hardy-Weinberg equilibrium is really one of the concepts that I think is really interesting about genetics. It's a mathematical approach to looking at the characteristics of a population. So this focuses on how allele frequencies would remain the same if certain requirements are met. So in order for Hardy-Weinberg to actually work or to be accurate, these requirements on this slide would have to be true. So no one would migrate, people would mate randomly, the population is extremely large, everyone has children, and there are no mutations. So we can see that this would not really, it's not a realistic view of the way the population is. It is a mathematical equation, and it focuses on how the allele frequencies would remain the same. It's we'll see the mathematical equation will be here on one of the other slides, but it's really kind of interesting. So if you're staying in the Hardy-Weinberg equilibrium, it's not really much fun. It's not really possible because everyone would have to stay in their same geographic region and no migration, and we know that that is really something that we can't do. So why is this important? So doing calculations to see if a population is in Hardy-Weinberg equilibrium can help scientists determine if evolution has occurred by documenting the changes in the allele frequency. So that's how this all came about, was that looking to see, scientists wanted to see if actually evolution had occurred. So this is why this is used. The founder effect. The founder effect says that if a small group of people leave a large population and settle someplace else, they take all their alleles with them. That makes sense. So an example would be Dutch settlers in South Africa and Huntington's disease. So you have a higher rate of Huntington's disease in Afrikaners because those Dutch settlers have a higher rate. So they took that all with them. So that's founder effect. The small population are the main people who populate the area, so that would make sense that they would have the alleles. Random mating, no selection based on traits or anything else. Assorted mating is purposeful selection of mates based on similar or dissimilar traits. I like to think that's kind of what the human population does. No mutation would be mutation increases genetic diversity, so if you don't have any mutations, your genetic makeup stays the same. Changes in the genotype over time allow for adaptation to the environmental changes. It also changes the frequency of alleles in a population, and it ruins your Hardy-Weinberg equilibrium theory. So, development of the Hardy-Weinberg equation, P is the frequency of the dominant allele, so we'll call that capital B. How many capital B's are there in a population? All the alleles in people who are homozygous dominant and half of the alleles in the people who are heterozygous, and that's the equation. Q is the frequency of the recessive allele, so the small b, and how many small b's are there in a given population. So all the alleles in people who are homozygous recessive and half the alleles in people who are heterozygous, which is the capital B small b, and there's the equation for that. The sum must equal 100%. So if there are only two alleles in the population, we can say that P equals 1 minus Q. The chance of all possible combinations of alleles in a population is P plus Q squared equals 1, or P squared plus 2P times Q plus Q squared equals 1. Listening to this, or to me voice over this, is not going to make a lot of sense to you. So I looked up on Khan Academy. This is where I get some of my genetic information. Khan Academy has a really nice video that goes over the equation and talks about, it uses eye color alleles to go through the Hardy-Weinberg calculations. So if you are so inclined, you go to Khan Academy and type in Hardy-Weinberg, and they will have this video, and it has to do with eye color. It's awesome, and it really will make sense to you much more than my voiceover on these slides. Genetic drift, moving on to a different concept, genetic drift is the process by which allele frequencies decline because of sampling error. Future generations are more like people who had lots of children than people who had few children. So the more children had by a population, the future generations are going to have those sorts of genetic traits. The trait is drifted out if there is less genetic diversity, if that makes sense. So in your textbook, there's a really nice example of this with drawing marbles out of bags, and you can see how it becomes diluted over time, that the traits become less and less diverse as you draw out the marbles from starting a 60 to 40 bag of marbles, so 60-40 split in terms of the trait. A population bottleneck is a little bit different. It's reduces the genetic diversity after like a natural disaster. So they can be caused by, like say, the Black Death. Anything that would reduce the population significantly can cause a bottleneck. Floods, famines, wars, Black Death. The result is that the population becomes less diverse because there's less genetic variation in the remaining population. So only the traits will be passed on from those who were survivors after this big, tragedy or whatever, disaster rather. So here's another marble scenario with a population bottleneck and drawing out you can see how when you're reduced down, the genetic variation is going to be really small. So future generations are going to have fewer diverse characteristics. Race and ethnicity, these categories kind of overlap. Racial categories overlap. Populations share common traits, but there really aren't sharp boundaries, and we can't use genetics to separate people into unique groups. People whose ancestors come from the same region do share the same alleles, but there's still some variation. Racial groups have genetic differences between individuals in a sociocultural group called races. It's much smaller than the genetic differences within each of these races. So ethnicity, more people identify with each other and believe that they share a common cultural heritage. It also takes into consideration human migration patterns, founder effects, and genetic drift. Categories of race and ethnicity, high-risk disorders and health disparities, and we don't really have an accurate way of describing people whose ancestors come from the same geographic region. So really looking at DNA doesn't really classify race and ethnicity as we know it culturally, if that makes sense. So this is the end of this slideshow. There is some additional things on the Blackboard page with some other resources, and I would encourage you to look at those as well and let us know if you have any questions. Thanks for your attention.